Welcome to PhilSci Hub Research University, your ultimate guide on your next research project.
We will lay down the best practices in research ideation, literature review, experimental design, and data analysis to guide you as you carry out your research project. Learn from the PhilSci Hub team of research scientists and some invited subject matter experts. PhilSci Hub Research University will be your personal coach through every stage of your research project, from ideation to final assessment.
And the best part? It's absolutely free.
Visit us at philscihub.com and be part of this learning journey.
Let's learn and succeed together here at PhilSci Hub Research University. Welcome to PhilSci Hub Research University, your ultimate guide on your next research project.
We will lay down the best practices in research ideation, literature review, experimental design, and data analysis to guide you as you carry out your research project. Learn from the PhilSci Hub team of research scientists and some invited subject matter experts. PhilSci Hub Research University will be your personal coach through every stage of your research project, from ideation to final assessment.
And the best part? It's absolutely free.
Visit us at philscihub.com and be part of this learning journey.
Okay. Can we get the slides on? Okay.
Magandang umaga po sa ating lahat. Welcome to the Filipino Science Hub. Thank you for your continued support to this online platform. To our new participants, thank you for checking us out today, and we would like to officially welcome you to the PhilSci Hub family. So bago po tayo magsimula, I would like to share some house rules in order to facilitate a smooth session for everyone. For our Zoom participants, we would appreciate it if you could turn your audio and video cameras off during the webinar. If you have questions or comments, feel free to type them on our Zoom chat box or YouTube live comments, and we will try to accommodate as much questions and comments as possible. After the talk, we will have a 30-minute Q&A where the Zoom participants will have the opportunity to ask their questions live. We would also like to request for everyone to not record the webinar. The full video of the session will be posted on our official YouTube channel. For those who would like to receive a certificate of participation, a Google form link will be posted on our Zoom chat box, YouTube live comments, and YouTube video description towards the end of the webinar. Please make sure that you enter the correct information, especially your email address, to ensure receipt of the certificate. Also, for you to obtain the certificate, you will need to subscribe to our YouTube channel as well as follow our page on Facebook. Please note that we receive hundreds of requests and it takes around 7 days or so to process all the certificates. If you do not receive your certificate after a week, please feel free to send us a message on Facebook or email us at philscihub@gmail.com. So again, welcome po sa PhilSci Hub. Welcome to all our Zoom participants and our YouTube live viewers. For those who are new and may not be familiar with who we are and what we do, allow me to share our mission and vision in the next few minutes. Filipino Science Hub is an online platform founded by our CEO, Filipino scientist Dr. Jeffrey Camacho Bungkin, back in 2012. Currently, We are a registered nonprofit organization based in Houston, Texas, United States. Our mission is to promote STEM education and the culture of research among students and teachers in the Philippines and abroad. Our current global leadership team is comprised of volunteer Filipino scientists and seasoned STEM educators from different parts of the world. Our vision is to see a technology and innovation-driven Philippines, and we believe that this can be achieved by creating a new generation of well-rounded Filipino STEM enthusiasts. This vision is being carried out via our two major programs, the PhilSci Hub Ed program and PhilSci Hub Research University. Through PhilSci Hub Ed, we are able to empower and support our teachers when it comes to strengthening STEM fundamentals education. We believe that this is important because the more equipped our STEM educators are, the more they can inspire our students to pursue a career in science, technology, engineering, and mathematics. Through this program, we have been able to serve our teachers by creating STEM teaching modules as well as conducting teachers training webinars, all of which are accessible on our website for free. So we have also been able to collaborate with major educational organizations that have the same goals and advocacies as Filipino Science Hub. Through these partnerships, we have been able to further expand our reach and help more students and teachers, especially during the time of pandemic. So together, everybody achieves more. The other major program that drives our vision forward is PhilSci Hub Research University. We believe that in order to strengthen a STEM culture, students also need to be able to learn how to conduct a scientific research. As we know, many scientific and technological breakthroughs happen through research, and we would like to equip our younger generation by providing them proper scientific research training. So ika nga po, start them young. Here at PhilSci Hub Research University, we provide free courses on fundamentals of scientific research, such as research ideation, literature review, how to write a research proposal, how to communicate your research work to the scientific community, among other courses. Through this program, we are also able to bring practicing scientists closer to the STEM educational sector. So because of this closer contact, our students become more aware of the possible STEM career paths available for them and how these careers contribute to the advancement of the society around them. So up to date, we have been able to reach over 70,000 students and teachers all over the world, and we know that this will continue to grow in the years to come. And this is why we are all very excited on what is in store here at PhilSci Hub. Our official website is www.philscihub.com. This is where you can access all our materials such as webinars, tutorials, modules, virtual lab, and special features. We are also present in several social media platforms such as Facebook, LinkedIn, YouTube, and TikTok. So follow us wherever you're most active on to get updated with our web events. So again, thank you for being with us this morning. And to officially start our webinar, I would like to turn the virtual floor over to our host for today.
All right. So magandang umaga po sa mga participants natin. Here on Zoom right now we have 40 participants, live participants here on Zoom, and also on YouTube Live we have 5 concurrent viewers as of now, so madadagdagan pa po yan later. And good morning everyone. My name is JP Onya. I'm one of the leadership team here at Filipino Science Hub, and I would like to welcome you to another webinar. This time about multi-species cytogenetic analysis, and this will be delivered by Mr. Michael Arnante of the Salle University Central Instrumentation Facility. Ayan, so this is part of the webinar series on modern analytical methods offered by DLSU CIF, and the team of CIF is very generous enough to offer this webinar series. And they're here right now, represented by Dr. Karen Lacerna, and of course, our speaker for today, Mr. Michael Ernesto Arnante. So good morning, guys.
Good morning, JP. So, good
So
morning,
happy to
everyone.
see you guys. All right. So again, welcome po sa mga participants natin. Some people are still coming in. So ayan po. So before anything else, no, we would like to introduce our facility sa DLSU sa ating mga participants. So, and to do that, I give the virtual floor to Ms. Karen Lacerna, Dr. Karen Lacerna of the De La Salle Central Instrumentation Facility.
Okay, so
Okay.
let me just share my screen. Okay, so again, good morning, everyone. So we're very happy to be here this Saturday morning to share with you about dun sa topic ni Mike. But before that, let me just introduce to you yung facility namin. So we are part of the De La Salle University Central Instrumentation Facility. So this is under the Office of the Vice Chancellor for Research and Innovation of De La Salle University. And we house a diverse range of advanced analytical instrumentations. So this is our building, the George SKT Advanced Instrumentation Building at the DLSU Laguna Campus in Binyan, Laguna. So sa mga taga Laguna dyan, sa taga Binyan, Santa Rosa, we're very near. So if you have any service needs or research needs, then you can just submit your samples ng madali, malapit lang. So we are established in 2019. We offer analysis, training, consultation, services, as well as we the research outsourcing. So yung mga research contracts, we also accept those kinds of services. But aside from these, we are very much open to collaborations and partnerships. So these are the different laboratories of Central Instrumentation Facility. So for your analytical needs, for your analytical requirements, we have our NMR lab. So we have a 600 megahertz NMR with solution probe and solid probe. We also have various spectroscopy instruments in the spectroscopy lab. We have a UVs with reflectance, FTIR with ATR, particle size, and zeta potential analyzer. We also have an ICP-MS for elemental analysis as well as an elemental analyzer. For our chromatography lab, we have QTOF LCMS, an analytical HPLC with UV, RID, and RID detector. And we also have capabilities for gel permeation chromatography for polymer analysis. We have a preparative HPLC with fraction collector and a GCMS with headspace autosampler. In the integrated electron microscopy lab, we have a field emission scanning electron microscopy instrument with energy dispersive spectrometer. So we have the imaging capabilities, as well as if you need yung element mapping for your included in the imaging. So that can also be done with this instrument. So yung EDS allows us to do that. We also have a transmission electron microscope and focused ion beam. We also have the imaging and cell culture lab. So this is yung lab nila Mike, so ICEL. So they have a confocal scanning laser microscope with live cell imaging as well as cell culture facilities. And lastly, we have in progress yung establishment of the wind tunnel testing laboratory. So they will have an Eiffel type wind tunnel equipment. So if you have if you need any of these instruments, again, we do not serve we serve not only De La Salle University, but we are very much open to serving the research needs and analysis needs of the rest of the country. So we are open to service analysis as well as to research and collaboration partnerships. Okay, so these are other equipments available at the lab. So if you have needs for sample prep, freeze dryer, rotary evaporators, as well as the other facilities and equipments, please do also contact us at cif@dlsu.edu.ph. Okay, aside from that, so now speaking of partnership, we are partnering with PhilSci Hub in this webinar series, Modern Analytical Methods. So in the past few months, you have heard from my colleagues, from Mr. Chris Argamino, talking about atomic and molecular spectroscopy for environmental research. In October, we had Dr. Jose Esmeria Jr. talking about fundamentals of scanning electron microscopy. Last week lang, we also heard from Dr. Virgilio Ebajo Jr. On the use of NMR for natural products research. And today nga po, we're having Mr. Michael Arnante speaking on multi-species cytogenetic analysis. And next year, I will be presenting on liquid chromatography and its application in natural products research. Okay, sorry. Again, this partnership is very much, we are one with PhilSci Hub in this partnership to promote education and research in the country. We hope that through this webinar series, we are providing our youths, our students, our graduate students, researchers, and teachers with the foundational knowledge on these various advanced analytical instrumentations and how can they be used for their basic and applied research. So we also wanted to share about how can we prepare our samples kasi of course it's important na to have good results, kailangan we also know how to prepare our samples well. So in this webinar series and most likely in the coming webinars that we will have with PhilSci Hub, we will be sharing about the foundational knowledge as well as how do you prepare samples to be sent to us, so that you can ensure that you can also get good analytical results. So with that, again, please do contact us if you have your service analysis needs, as well as if you would like to establish collaborations and partnership. Email us at cif@dlsu.edu.ph.
All right. Okay. So thank you, Karen. So nabanggit ni Dr. Karen Lacerna na webinar series po ito, and this is now the 4th installment of this Modern Analytical Methods webinar series. And kung na-miss nyo po yung mga past na webinars, lahat po yan ay recorded, no? So makikita po natin lahat ng recorded videos sa website po ng PhilSci Hub at www.philscihub.com. Okay? So, ayun. So right now, we would like to present or introduce our speaker, and I would also like to call on Dr. Karen again to introduce her colleague sa CIF DLSU.
So
Si
yun,
Sir
okay.
Michael Arnante. All right.
Yes. So again, it's my pleasure to introduce Mr. Michael Arnante, my colleague at the DLSU CIF. So he is the equipment specialist of the ISA laboratory. He had his MS in biology from the University of the Philippines, Diliman. He is currently having his PhD in biology at the Salle University. He was a former technical assistant in the molecular cytogenetics laboratory of the Research and Biotechnology Division of St. Luke's Medical Center. Currently, he is in charge of the Imaging and Cell Culture Laboratory, housing confocal laser scanning microscope. So with that, let's join me in welcoming Mr. Michael Arnante, sharing about multi-species cytogenetic analysis. So good morning, Mike.
Okay, Sir Mike, the floor is yours.
All right. Magandang umaga po. Good morning, everyone. Thank you again to PhilSci Hub for this invitation, this webinar. I hope I'll be able to share my experiences and my knowledge in terms of cytogenetic analysis. So, sige, let me just share my screen. Sir JP, pa-share lang po ng screen. I think na-disable yung co-host. It's okay?
Wait a second.
All right. So I hope
Okay.
everyone can see my screen, no? So
I can see it now.
all right. So ayan. So again, good morning everyone. Magandang umaga po. I'm here to talk about today about multi-species cytogenetic analysis. Now, personally, no, in cytogenetic analysis, most of my work previously involved cytogenetic analysis for human samples, since I previously worked in a hospital. So most ng experience ko po sa cytogenetics is really dealing with patient samples. Now that I'm currently working in an academic institution, I will be working mostly na with animal and plant species. Meron pa ring human, no? Meron pa ring human samples. I'll show you how we can still make you still work with human samples as we go on with this lecture. Okay? So to start, just in case some people don't know, no, I want to share what is cytogenetics. Okay, so to define, cytogenetics is a field of genetics centered on the study of the structure and behavior of chromosomes. Okay, so ano ba ang chromosomes? Ang chromosomes. So chromosomes carry an organism's genes and therefore majority of its genetic information. Okay? So nandito po sa chromosomes ang ating genes, is the physical structure ng ating DNA, no? So some of us may only know DNA as the double helix structure, di ba? Yun yung familiar tayo, di ba? You have your double helix structure and then you have your bases, your adenine, thymine, cytosine, guanine. That's the molecular structure of your DNA. But in the cell, no, you'll be able to visualize your DNA in the form of chromosomes. That's why the field of cytogenetics po is actually the observation, no, the observational study between the cellular and the molecular level. There are some things, no, there are some factors na makikita lang natin pag cellular, and then there are also some things na makikita lang natin when we do experiments on the molecular level. But cytogenetics actually is the bridge between that. Okay, you can do studies on a molecular level, but will be able to visualize it under the microscope. Okay, syempre with specific factors, no. So yun, that's what's interesting about the field of cytogenetics. Okay, so this is just a small, simple diagram, no, of what exactly are chromosomes. What do we look at when we do cytogenetic analysis? So you have your cell, di ba? Basic biology, you have your cell, you already know what's in your cell. Now inside your nucleus, that's where you can see your chromosomes, di ba? And then your chromosomes, how they are structured, you have your condensed chromatids, di ba? And then your chromatids are composed of nucleosomes. Nucleosomes is the unit, no, the packed unit of your DNA, which include your DNA wound around proteins called histones. Okay? And then yung DNA na yun, syempre, alam na natin yung standard structure ng DNA. You have your double-stranded DNA, sabi ko nga kanina, in a double helix form. And then you have your base pairs. Okay? So as you can see, no, sabi ko, I always reiterate, no, it's the bridge between cellular, you can view it in cells, and the molecular level where you can do your base pairs. So we visualize chromosomes in that aspect. Okay? Now, how do we visualize chromosomes? Yun yung i-discuss talaga natin today. Now, the most basic form, no, the most basic form of cytogenetic analysis is called karyotyping. Right? So karyotyping is defined as the preparation of an organized profile of an organism's chromosomes. Okay? So this is how we arrange. So under the microscope, syempre, no, when you visualize chromosomes, hindi pa yan arranged. Okay? So the process of analysis and doing that is called karyotyping. Now, karyotyping is essential for detecting chromosomal abnormalities in whatever your sample is. Actually, hindi lang chromosomal abnormalities, no? You can actually determine the state, you can determine the length, kung gaano kalaki yung gene component ng isang organism, no? And then, you can actually detect the presence or absence of genes. Sometimes it's not necessarily an abnormality, no? You just want to see if the gene you're looking for is present or absent, okay? In a visual component. Of course, you can do that in a molecular manner, no? Pwede kayo mag-PCR, di ba? Pero if you want to visualize it, you do cytogenetic analysis or karyotyping. Okay. Now, the most common, sabi nga ni Doc Karen kanina, no, very important yung knowing how to do sample processing. Okay. Now, in processing samples for karyotyping, at least on my experience, no, the most common way of processing samples for karyotyping is your blood culture. Syempre, on organisms lang which you have blood, di ba? Like in humans or in some higher animals like monkeys, cattle, cats, dogs, or sino pa, basta any organism that has blood, no? You can actually do blood culture for karyotyping. Now, that blood culture, specifically, no, is called short-term lymphocyte culture. So remember, lymphocytes. Bakit lymphocytes? Lymphocytes are white blood cells, no? We know that our red blood cells, wala silang nucleus, no? They don't have any nucleus. Therefore, walang DNA, walang chromosomes. Right? So, yun lang yung kinukuha natin when it comes to chromosomal analysis. We do processing of lymphocytes. Okay? So now, in culturing blood, very easy, no? If you're able to extract blood, you can culture it right away. Syempre, in terms of processing, syempre may ano din tayo dyan. Meron tayong rules, di ba? Especially when you're dealing with infectious samples. In some cases, all blood is considered technically infectious, no? May mga blood-borne diseases tayo. So be very careful when you want to process your blood samples. I think ang requirement ngayon for processing blood samples is at least a BSL-2 level biosafety, no? So if you do want to process your blood samples in your university or institution, make sure na meron kayong ano din, no? Meron kayong facility na appropriate for blood cultures, okay? So yun. In doing lymphocyte cultures, the standard way of processing is you have your standard media. Syempre, lahat ng culture kailangan ng media. So we use RPMI 1640. That's the standard media for blood cultures. You have your fetal bovine serum as a nutrient source, and you have your penicillin streptomycin to prevent contamination. These are antibacterial, antifungal agents, no? Siyempre, ang gusto nating i-analyze is yung sample galing sa organism of interest natin. We don't want to see contamination by fungi or bacteria. And then, what's important in doing blood cultures, no? We can actually manipulate them to grow and proliferate in our culture. Okay? So, we add what we call an agent called phytohemagglutinin. So what phytohemagglutinin does is it induces mitotic activity. Remember cell division in circulating blood, no? Yung blood na nag-circulate na sa katawan natin, it doesn't undergo cell division, di ba? Your cell division ng blood cells only occurs in the bone marrow. Okay? So pag blood sample ang kinuha natin for karyotyping, we have to induce mitotic activity para mag-multiply yung blood cells natin. So that's where we add phytohemagglutinin. Now take note that you can actually do karyotyping in bone marrow samples. We used to do that, no, when I was still working. Since we're working with cancer cells, in cases of leukemias, no, we want to see the genetic abnormalities at the source. So, di ba, in human patients, syempre ang source natin is yung bone marrow. Alright? So yun, pero pwede din, di ba? But again, it's much more difficult to get a bone marrow sample, syempre, no? We want to see blood sample is much easier, right? So after culturing, na-culture na, no? Pag na-culture na with the standard, after about 48 to 72 hours, you do now your harvest of lymphocytes. Okay, so kukunin na natin yung sample from the culture. Another important part of processing samples for cytogenetic analysis is the addition of this reagent called colcimid. Okay, now what colcimid does is it prevents formation of spindle fibers. Right? So why do we need to prevent formation of spindle fibers? Because we want to lock cells at the metaphase stage of the cell cycle. Okay? We go back into our high school biology, di ba? Sa mga high school biology, tinuturo sa atin lagi, di ba? Yung stages ng cell cycle, or specifically the stages of mitosis. So this is an example, no? So you have your cell cycle, di ba? So you have your G0 or your interface wherein your cells grow and develop, no? And then at a certain point, it reaches, it starts to undergo mitosis. Now your mitosis have different stages. So your prophase, metaphase, anaphase, telophase, and then when it becomes daughter cells. By the way, may prometaphase na pala ngayon. That's another stage of the cell cycle. Now, as I said, in doing cytogenetic analysis, we want to lock cells at the metaphase stage, no? Here, the metaphase stage of the cell cycle. Now, why do we need to do that? Remember, in prophase, at the start, no, the start ng mitosis, your chromatin material, so in the cells, no, start to condense. Dito lang na actually nagsa-start mabuo yung chromosomes. Okay? In normal stage, in normal growth and development, no, in interphase, ano pa yan? Widespread pa sila. Hindi pa sila buo. Yung chromosomes natin are not in their most intact form. It's only in prophase wherein they start to condense. Okay? Now, in metaphase, the next stage, no, ng cell cycle, remember what happens during metaphase. Your chromosomes align at the middle of the equatorial plate, no, it's called the equatorial plate in cells, no, and then they align, and then they are attached in their centromeres by the spindle fibers, right? And then during anaphase, pag nag-progress na siya sa anaphase, no, remember, your sister chromatids will now split. Okay, they will split, and then they will move toward opposite poles. Okay, so yun yung nangyayari sa anaphase, and then in telophase, magsiseparate na sila, and then that's when you have your daughter cells. Now, remember what colcimid does. It prevents formation ng spindle fibers. So remember, your spindle fibers is what pulls them toward opposite poles. Now, since walang nag-form na spindle fibers, your cells will not continue now to anaphase. Ibig sabihin, lock yung cells at the metaphase stage ng mitosis. And then dahil yun, at the metaphase stage na, yung pinaka-condensed form, yung pinaka-visible form ng chromosomes natin, ano? So doon natin ma-visualize in its best state yung ating chromosomes. That's why we lock them sa metaphase stage ng cell cycle. Okay? So yun yung very important when you want to do processing of samples for chromosome analysis. Okay? So, before we go into the staining, part ng ating chromosomes, so the next step kasi is staining, no? I want to talk shortly about sample processing. As I said, the most common is blood culture, no? Most common. But then again, not every organism has blood. Or sometimes we do not have the right facility, no, to do processing for blood samples. Maraming ways. There are a lot of different ways we can do processing for chromosomal analysis, no? The easiest and probably best way to do this is to do processing for cell culture. There are commercially available cell lines in the market. So, pwede kayong bumili na ng process ng sample, cell culture. Siyempre, may sarili pa rin kayong processing, no? But it's easier to do. And if you do process commercially available cell lines, alam ninyo na controlled yung conditions nun. So it's much better for your experiments to do that. So as I said, there are commercially available cell lines, no, even human cell lines. Maraming, when we do cancer studies, no, when we do cytogenetic analysis for cancer studies, maraming human cancer cell lines na nabibili na sa market natin. So if you want to test, for example, no, if you want to do tests on a specific type of cancer, if you want to do anti-cancer studies, no, marami nang nabibiling human cancer cell culture lines. Okay, so you can do that para hindi na kayo magpa-process from a raw sample. Animal cell lines as well, para kung sakaling you don't want to process samples from a living animal, no, from a living animal, maraming nabibili din na animal cell culture na, and plant cells as well. Okay, so it's not necessarily blood cells. Maraming samples na pwedeng i-process for cytogenetic analysis. Now, if you really want, no, if you really want to do samples from, let's say, from the field or from your own population, from your own interest, meron pa ring ways to do that, no? Like for plants, for example, if you do want to do cytogenetic analysis in plant samples, so maraming ways. There's the squash technique, no? If you remember, very basic experiment sa high school biology natin is yung when we process onion root tips. Diba? Tinitingnan natin sa slide if we want to view cells undergoing mitosis. Diba? Very basic experiment yon sa high school biology natin. I-modify lang natin ng konti yun. I-modify lang natin ng konti yun. And then you'll be able to process samples from plants to do cytogenetic analysis. Take note lang na as long as your samples are from the apical meristems. Ano yung apical meristems? Tandaan natin, apical meristems are either the shoots or the roots ng plants. Most samples doon ng cytogenetic analysis, as far as I know, no, it comes from root tips. Okay? So yun kasi yung madaling i-process, yung root tips. So you'll be able to visualize your chromosomes from the root tips. Now, in some organisms na walang blood culture, no, a prime example of that is from insects. Okay? You can actually do chromosomal analysis from insect samples as well. They do the smear technique, ganun din. Kinakrush lang nila. Sometimes in large enough insects, no? Sometimes in large enough insects, they extract specific glands. Glands, no, ng insects. I think ang alam ko is pwedeng salivary glands or the gonads, the reproductive organs ng insects, para to do chromosomal analysis. So kinakrush nila yon, then sinismear lang nila along with some fixatives. Okay? So pwede, no? Maraming paraan ng sample processing. You just have to do enough research to know kung ano yung tamang pag-process ng samples. Alright? So with that said, okay na tayo, no? Na-process na natin, na-isolate na natin yung lymphocytes. In case of other samples, depende dun sa cells na hinahanap natin. But we go back to blood culture, no? Let's say we go back to blood culture, we process the lymphocytes. What would be the next step? The next step is now to stain your chromosomes para makita natin siya under the microscope. Okay? So the most common method of staining chromosomes for differentiation is called the GTG banding technique, or Gbanding for short. The GTG stands for G-bands by trypsin using GEMSA. Now, it is the most common method of staining chromosomes for differentiation. All right? Now, GEMSA stain is very common. Madali lang. Actually, in schools, pwede kayong gumawa ng GEMSA staining. Readily purchasable naman ang GEMSA stain, no? But if you do simple GEMSA staining, right? If you do simple GEMSA staining lang, yes, you will be able to stain the chromosomes, no? Pero hindi nyo ma-differentiate kung which chromosome is which. Remember, each chromosome, may chromosome number yan, di ba? May chromosome number yan. Now, in order to visualize our chromosomes, no, para malaman natin which chromosome is which, you have to have what we call a differentiation technique. Okay? So makikita natin under the microscope, pag in-stain nyo ng GEMSA, yung chromosomes nyo, no, lahat sila, isa lang itsura, yung X. Nag-iiba-iba lang yung sizes, no? Yung parang X ang itsura ng chromosomes natin. Iba-iba silang sizes, iba-iba silang centromere position, but that's it. Hanggang dun lang yung kaya nating i-differentiate, no? But when you do G-banding, you'll be able to visualize each chromosome because they will exhibit specific alternate light and dark bands. Okay? Within the chromosome, mabivisualize yun na may specific alternate light and dark bands. Now, how does that happen? G-bands by trypsin work by the digestion of chromosomes by the proteolytic enzyme trypsin. Some regions of the chromosome kasi, no, get digested faster than some regions. Depende sa ratio ng adenine, thymine, and guanine cytosine content. Alright? So some regions will not take up the stain. So gems stain will still provide your color differentiation. No? So some regions will be lighter stained and some regions will be darker stained. So magkakaroon kayo ng pattern, magkakaroon kayo ng identifying pattern in which malalaman nyo, magagamit nyo siya to know which chromosome is which. So after G-banding, ganito yung magiging itsura ng chromosomes niyo. So parang ganito. Right? So under the microscope, this is what they look like. So kung titingnan nyo, no, each chromosome, hindi lang siya isang dark stain. Meron siyang alternate light and dark banding patterns. Okay? So sa humans, ganito yung itsura niya. No? Sa animals, I think this is either a mouse or a deer chromosome. Forgot lang, no? I think this is one of those animals. So ganun din, no? Meron din siyang pattern. Meron din siyang alternating light and dark patterns. Okay? Now, because of these alternating light and dark patterns, you'll now be able to identify, malalaman nyo na ngayon kung anong chromosome number yung target ninyo. Okay? Now, when I was only starting in this field, no, what we used to do was, one of the primary tests in being a cytogenetic analysis was dapat sa isang tingin mo lang, alam mo na kung anong chromosome number na yun. So we were actually required to memorize the banding pattern of each chromosome. Now, syempre at the start, medyo mahirap, no? May kodigo pa kami noon. We have a guide. We used to have a guide on which banding pattern. Now, at least, no, ako, kasi nga since ito yung field ko talaga, no? So kaya ko nang i-identify which chromosome is which. For example, no, if you can see my pointer here, no, as a guide, this is chromosome X, this is chromosome 5, this is chromosome 1, this is chromosome 12, this is chromosome 10, this is chromosome 21. So that's part of the skill of cytogenetic analysis, being able to identify which chromosome is which because of their banding patterns. Okay. Siyempre, if you want to work on cytogenetic analysis, pwede naman namin kayo turuan. Eventually, we can actually teach you on knowing which chromosome is which based on the banding patterns. Yun. Pero okay lang mag-kodigo, syempre. Di ba? Okay lang yun. There's no harm in doing that. As long as you correctly identify which chromosome is which. Okay? So, yun. So, this is what you do. So, okay na, di ba? Na-visualize nyo to. You're able to visualize na your chromosomes under the microscope. What do you do next? You now do your analysis. Okay? You now do your analysis. So, your analysis, you want to arrange your chromosomes in pairs. Most naman ng chromosomes natin, especially in diploid organisms, we humans are diploid organisms, no? We will be able to do your chromosomes in pairs. Diba? Syempre, we have our 2 copies of each chromosome. Now, chromosomes are arranged according to size. So kung gaano kalaki yung chromosome, banding pattern, like I said, no, may sinusunod tayong banding pattern, and your centromere position. I'll discuss later yung centromere position. No? So your chromosomes are based now on what we call ideograms. Okay? Ideograms are published identified guides na on chromosomes of each species. Diba? Most naman ng species ngayon, may ideograms na. Since we're gonna be talking about multi-species today, no? So in humans, for example, ito na, may identified na banding pattern na dapat yung chromosomes natin. So ideally, we'll be able to see most of these bands, no? Most of these bands when we do look under the microscope in chromosomes. Okay? Siyempre, not all the time we'll be able to see these bands. No? Depende sa condensation level ng chromatin natin. Sometimes we'll have short chromosomes, sometimes we'll have long chromosomes. And the longer chromosomes you have, if you're able to catch your chromosomes at a longer condensed state, no, then you'll be able to see more bands. Okay? But part of the skill in doing cytogenetic analysis, no, is whether your chromosomes are long or short, dapat alam mo pa rin yung notable na bands sa bawat chromosome. Okay? So yan. So for example, no, ito yung guide natin sa humans. In other animals, no, marami na ring published na ideograms. For example, in dogs. So dogs, alam natin, which they have 78 chromosomes. So ang dami, no? Meron na ring ideogram yan. So when you do Gbanding of dog chromosomes, malalaman mo rin which chromosome is which because of the banding patterns. Okay? So meron ng guides, marami namang guides na on the chromosome banding patterns of different species. Right? So siguro tingnan niyo lang kung ano na yung published data, on your organism of interest. Okay? So that's an analysis. In terms of classification, I talked about centromere position kanina, no? So the centromere is actually the central constriction ng inyong chromosome. Diba? When you're visualizing chromosomes, parang X ang itsura nila, diba? Yung centromere is yung middle point ng X na yon. Okay? And in different chromosomes, iba-iba yung position ng centromere na yon. So you'll be able to use the position of that centromere as a guide para malaman din kung anong chromosome yung tinitingnan ninyo. For example, specifically itong guide na to in human chromosomes, no? So for example, chromosomes 1 to 3, we have large chromosomes and they are metacentric. Metacentric means that your chromosome is in the middle. Your upper arm, the upper arm is called the P arm, and the lower arm is the Q arm. It's called the Q arm, no? So in metacentric chromosomes, the length of the P arm and the Q arm is almost equal. Okay? So halos same size sila. Okay? So yun yung metacentric. So we know that chromosomes 1 to 3 in humans are metacentric chromosomes. Chromosomes 4 to 5 naman, they are known as large chromosomes relatively, but they are submetacentric. Submetacentric chromosomes are when your P arm or yung upper arm is shorter than the long arm or the Q arm. Okay, mas maiksi yung upper arm ninyo. So sila yung submetacentric chromosomes. Okay, the third type naman, no, is called you have your chromosome 6 to 12. Submetacentric din pala yan, by the way, but they are short, they are medium size along with the S chromosome. The third type starts with your chromosome 13 to 15. So they are medium size, but this time they are acrocentric. Okay? Acrocentric is halos wala na yung P-arm ninyo. Diba? Almost satellite DNA na lang, almost satellite chromosomes na lang yung nasa P-arm. Okay? So these are what we call acrocentric naman ng chromosomes. There's one more type, no, what we call the telocentric. The telocentric chromosomes as in wala na talaga. Absent na talaga yung P-arm, but we humans do not have that telocentric chromosomes. No? Some organisms still do have that. Okay? So as we said, no, size din ang isang basis ng identifying chromosomes. Kaya nga we have your relatively large chromosomes from 1 to 5, medium-sized from 6 to 15, and then your 16 to 22, and your Y chromosome is small. Pero kung titingnan nyo, iba-iba yung centromere positions nila. So that's what we use, no, to identify your chromosomes. Right? So after analysis, pag na-analyze nyo na, no, na-analyze nyo na yung chromosomes niyo, na-identify nyo na which chromosome is which, the final result of doing your cytogenetic analysis is what we call the karyotype. Okay? Now, in humans, this is what you'll be looking for. If you have human samples ng karyotyping, this is what you're going to be looking for. So we know that humans have 46 chromosomes. And then magkaiba, no, pag males and females. So you have your males, which is XY. So you have 46 XY. And then you have your females, which is 46 XX. Okay? So now, this is the normal state ng human chromosomes. Now, any change from this, any change, when may maiba lang sa karyotype na to, that's what you consider now an abnormality. Okay? So yun yung hinahanap natin in doing cytogenetic analysis. Okay? So now, what kinds of abnormalities are we looking for? Right? So the first type is what we call the numerical abnormalities. So numerical abnormalities is the deviation from the normal chromosome number. So tayo, in humans, our normal chromosome number is 46. Diba? In other organisms, you have to know yung normal chromosome number nila in order to know kung ano yung abnormal number sa kanila. Syempre, di ba? Okay. Some examples of numerical abnormalities are when you have your monosomies. Monosomies is kung isa lang yung copy ng chromosome ninyo. Remember tayo in diploid organisms, di ba? We have 2 copies of each chromosome. Kung nawala yung isang buong copy ng chromosome number na yon, you have your monosomy of that chromosome. Trisomies naman is when you have an extra copy ng chromosome na yon. So instead of 2, you have 3. Okay? And then you have your tetrasomy. Sometimes you have 4 copies of your target chromosome. So yan, titingnan natin din yan later. You can also have your triploidy or tetraploidy. Diba? Triploidy is when you have 46, diba? 46 ang normal chromosome number natin. But that's diploid. Okay? So when your chromosomes become triploid, so you will now have 46 plus 23, you now have somewhere in the range of 69 chromosomes. Diba? And then tetraploidy is when you have umabot na, nag-multiply na, naging 4 times na. So you have 92, somewhere in the range of 92 chromosomes na. Now, this usually happens in cancer cells. Diba? One of the characteristics of cancer is rapidly dividing sila, di ba? Uncontrolled cell division. So sometimes in uncontrolled cell division, nangyayari yung occurrence ng triploid and tetraploid chromosomes. So that's why makikita na. So those are all numerical abnormalities. Nag-iba yung chromosome number. Okay? So the next type of abnormality is called your structural abnormalities. Now, structural abnormalities happens when you have changes in structure and morphology. Okay? Nag-iba yung itsura. Pwedeng 46 pa rin siya. In humans, no? In humans, pwedeng 46 pa rin siya. But may naiba, may naputol, may bumaliktad, may dumoble yung isang part lang, and then may nag-exchange ng material. So yun yung mga yon, deletions kung may naputol, duplications kung may nadobling isang part, inversions kung may nabaligtad, and then translocations kung merong nag-exchange, nagpalit ng material between 2 different chromosomes. So yun yung mga tinitingnan natin, yun yung mga different types of abnormalities. So some examples of abnormalities, no, very common. Some of these are we are already familiar, no. So very common, the most common human chromosomal abnormality is of course our Down syndrome. Yan, very familiar sa atin yan, no. So Down syndrome, you have your 3 copies ng chromosome 21. Okay. So, this is a typo error. XX pala dapat. Mali yung na-paste ko. XX dapat yan. So, this is a female. Typographical error. This is a female with 3 copies of chromosome number 21. Okay. So, some other syndromes, no? You have your Edwards syndrome. You have your Edwards syndrome, trisomy 18. And then you have your Turner syndrome, sex chromosome abnormalities, no? Now, in females, isa lang, dapat 2, di ba? Remember, dapat 2 yung copy ng X chromosome in females. In cases of Turner syndrome, isa lang yung copy nila. So these are cases of basically underdeveloped females. In males naman, so they have your Klinefelter syndrome or XXY. So dapat pag males, XY lang, di ba? So Klinefelter syndrome are may extra copy ng X chromosome. So these are basically the feminized males naman. Kasi naging 2 yung copy ng X chromosome nila. But they're still males though because of the presence of the Y chromosome. Okay. So these are just some of the abnormalities, no, in at least in humans, no? At least in humans. So these are some numerical abnormalities. Now when it comes to structural abnormality naman, one of the fields that I have experienced in working is in cases of leukemia, specifically chronic myelogenous leukemia. It is actually the most common type of leukemia. No? It is 20% of all leukemia cases. We know that there are lots of different types of leukemia. No? So CML, chronic myelogenous leukemia, is characterized by an overproduction of cells of the granulolytic and occasionally the monocytic series. Okay? So again, maraming medical maraming medical implications yan. But in terms of cytogenetics, in terms of chromosomal analysis, no, what's important to note is CML cases, in 90% of CML cases, no, you have the presence of what we call the Philadelphia chromosome. Okay. So ano tong Philadelphia chromosome na to? It's one of the perfect examples of a structural abnormality. So this is an example of a karyotype with a Philadelphia chromosome. Okay. So Philadelphia chromosome has what we call the reciprocal translocation between chromosomes 9, so chromosome 9, no, and chromosome 22. So there's an exchange. Nagkaroon sila, nagpalitan sila ng genetic material, nag-exchange sila. And that exchange is what triggered, no? Pag meron yang genetic abnormality na yan yung nagde-trigger sa body para magkaroon ng leukemia. Okay? So this specific genetic abnormality, okay, is what causes. So this is one of the examples of a cytogenetic abnormality. So this is cancer na, no, leukemia. So very important to take note. Okay. So other types, no. So that's for Gbanding. That's for GTG banding. Now marami pang other types of chromosome staining on studying chromosomes. For example, you have your C-banding. So C-banding only stains your centromeres. Okay? So kanina, ang gusto nating tingnan lang is yung banding patterns in Gbanding. Kaya siya pa rin yung one of the best, no? C-banding, kung centromere lang yung gusto tingnan natin. Some studies on plants kasi, no? Some studies on plants only wants to view the centromere position. Okay? Now, if you do want to take a look at that, is yun lang yung tingnan ninyo, yung C-banding, centromere lang. Another type is your R-banding. Your R-band is kabaligtaran ng G-banding. Alright? So it produces positive bands that correspond to the negative G-bands. Okay? So kung gusto nyong baligtad yung orientation ng G-banding ninyo, for some reason, no, there are some studies na gusto nila baligtad yung orientation ng bands nila. So that's why they do reverse banding. Okay, so specific, depende sa gusto ninyong analysis. But mostly G-band pa rin yung ginagamit na staining techniques. Alright? So marami pa ibang klase, but these are just the most common. Okay? Now, as you saw dun sa diniscuss ko about G-banding, that's very basic. Under the microscope, you'll be able to see light and dark banding patterns lang. That's well and good if you want a basic microscope. What if you want to see your chromosomes colored? Gusto niyo na makulay ang inyong chromosomes. So you now do a different technique for cytogenetic analysis, which is called FSH. Okay? So FSH stands for fluorescence in situ hybridization. Okay? So FSH uses fluorescent molecules to vividly paint chromosomes or just a particular gene. Kung gusto niyo isang gene lang ang kulayan niyo, pwede. Kung gusto niyo kulayan yung buo, pwede. It's really up to you. Yun yung advantage ng FSH, no? It's very specific if you want to study only a specific part of the chromosomes. Now, it is useful in disorders with very small aberrations or samples with no metaphase cells available. Very small aberrations when sometimes may mga genetic abnormalities tayo na very small and then hindi natin siya ma-visualize under the microscope under normal G-banding techniques, 'di ba? Kasi nga light and dark lang siya. Let's say for example may na-delete kayong isang small part ng chromosome, no? Pag na-delete 'yon under the microscope, hindi niyo makikita enough 'yon. Kung if it's too small, hindi niyo siya makikita. Whereas when you do FSH, you'll be able to visualize kung nandun pa or nawala na yung specific gene na hinahanap ninyo. Sometimes naman, as I said, no, in metaphase, you have in Gbanding, it's very important to get your cells at the metaphase stage. What if magkaroon kayo ng problem with cell division? Sometimes hindi nyo nakukuha yung cells at the dividing stage. Mahirap. And some samples, very difficult yun. When doing FISH or fluorescence in situ hybridization, kahit wala kayong dividing cells, okay lang. You'll be able to see your chromosomes or the genes at least, no, the genes na tina-target ninyo kahit walang metaphase cells available. So as I said, you can use FISH for more specific gene targets. Okay? So bakit? Paano natin nagagawa yun? FISH kasi uses complementary DNA sequences of target genes. So we're going here na into a bit more molecular. Okay? So we're still in the chromosome level, no? Pero we're at least venturing into the more molecular technique na. Okay? So sabi ko din, no, selective or whole chromosome staining. You can choose to color all chromosomes or you can choose to color just selected parts. Depende kung ano gusto ninyong aralin. So these are some examples of, this, by the way, ito pala yung principle, no? Before I go to the examples, this is the principle of how FISH works. So you have your sample DNA. Remember, the chromosomes are still made of the same DNA sequences. Okay? So yung DNA sequences na yon, you have your, alam na natin yon, lalo kung na-sequence na natin yung DNA, di ba? So we are ATCG sequences. So what you do is, you synthesize a complementary sequence of your target. Okay? So complementary, remember, your adenine is complementary to thymine, and your cytosine is complementary to guanine, and vice versa. Okay? So kung alam niyo yung DNA sequence niyo, madali nang mag-synthesize ng complementary DNA sequence. Now, if you have your complementary DNA sequence, what you do is you attach what you call a fluorescent probe. So it's a molecule that emits fluorescence under certain conditions. Okay? In the process of FISH, that certain condition is when your complementary strand attaches to your target DNA strand. Okay? Remember, your attachment only happens kung yung sequence ninyo is complementary. Diba? Kung tama yung sequence ninyo ng complement, doon lang siya kakabit. Kung hindi naman siya complementary, it will not attach. It won't happen. You will not be able to see fluorescence because of that. Okay? So hindi siya magiging visible under the microscope. Okay? So you can see, no, once the complementary DNA sequence attaches, so dun ngayon, makikita, dun ngayon mag-visualize yung fluorescence under the microscope. Whether it's direct label or sometimes amplified siya, no? If you want to see more fluorescence, direct label siya. Okay, direct label siya. Okay, so yun. So how does fluorescence in situ hybridization look like under the microscope? So ito na, makulay na siya, no? Kanina black and white lang. Kanina black and white lang yung samples natin. So this time, may color na yung sample natin. So as I said, selective, no? Selective yung kulay natin. For example, in this case, ang gusto lang nating kulayan, what we only want to color, no, is chromosomes 1, 2, and 3. Maybe that's our interest lang. That's why we want to color this, no? So you can see, no, we colored chromosome 1 colored green. Okay? And then chromosome 2, we wanted to color yellow. And chromosome 3, we wanted to color red. By the way, no, if we do want, we can actually assign colors. If we want to, pwede tayo mag-request sa gumagawa. Although there are commercially available FISH probes na, no, technically we want, kung gusto natin ng chromosome 1 ay kulay ganito, pwede natin igawa yun. Siyempre, i-request lang natin yun. But there are already commercially available FISH probes for that. So as you can see here, no, chromosomes 1, 2, 3 lang yung may color. All the rest are what we call counter stain. Ibig sabihin, it's the general stain. The normal counter stain is DAPI. Okay, and it will stain all your chromosomes fluorescent blue. Okay, so yun. So kaya ang usual stains ng chromosomes ay none other than blue, no? Kasi blue is the usual counter stain under the microscope. Okay, so that's an example. On the second image, you can see, no? So sabi natin kanina, I discussed about the Philadelphia chromosome translocation. The chromosomes between 9 and, the exchange of material between chromosomes 9 and 22. Now, if you can see here, no, ang kinulayan lang dito sa sample na to is chromosomes 9 and 22 lang. Okay? So chromosome 9 is supposed to be colored red. And then chromosome 22 is colored green. Now, if you look here, no, chromosome 9 is red, chromosome 22 is green, nag-exchange ng genetic material. Kaya may green na part yung chromosome 9 and may red na part si chromosome 22. Kasi nga, nagpalit ng genetic material. Your genetic material, wherever they go, no, it doesn't matter. It will be still that same color because yung target gene sequence nyo is assigned that color. Okay? So kahit saan mapunta yan, kung saan man siyang lumipat, mag-fuse with a chromosome, it will still stay the same color that you assign. Okay? Yun. So ito naman, the third image, sabi ko nga kanina, no, it doesn't need to be at the metaphase stage of the cell cycle. If you're looking for a certain gene, so in this case, same lang din yun, translocation between chromosomes 9 and 22. So that red and green color fusion is visible kahit na yung cell ninyo ay non-dividing. Okay? So it's very informative in this case, no? And then if you really want to extremely color your all chromosomes, pwede, no? So if you can see here, this is what we call a multi-band fish. Okay? So hindi lang yung chromosome single color. Each band, so sa Gbanding natin, alam natin may band yung chromosomes, di ba? Each band has a specific assigned color. Pwede yon kung gusto na talaga specific. Ang rule lang sa fish, no? Ang rule lang when doing fish, mas makulay, mas mahal. Okay? The more colorful your fish is, the more expensive it will be. Kasi nga, syempre, there's more fluorescent probes to be synthesized. Okay? So, yon. So, speaking of multicolor fish, no, as I said, in our case, we call that spectral karyotyping. Okay? So, it's multicolor fish resulting in a fully colored karyotype. Each and every chromosome of the organism is assigned a fluorescent molecule template, right? So there's a template, there's already a color or multiple colors assigned to specific chromosomes. So they can be whole chromosome probes, isang kulay lang bawat chromosome, or multi-band probes. So sabi nga natin, no, every single band makulay. Okay? So yun. So depende kung anong interest niyo. Right? So these are the different methods. These are the different methods of chromosomal analysis or cytogenetic analysis. Now, saan ba natin magagamit? Why do we need to do chromosomal analysis? Ano ba ang purpose? Para saan? What kinds of studies can we do cytogenetic analysis? Okay. So just some examples, no? So I want to start with anti-cancer studies because this is really my field of interest, no? Cancer studies. One of the examples of studies where you can do chromosomal analysis is when you're doing natural products research, right? So you have a specific extract from a specific plant or bacteria or fungi, kung ano man, no? Kung ano man yung sample ninyo. So you want to test your extract for possibly anti-cancer activity or not necessarily anti-cancer, no? What changes? You want to see what kind of changes your extract affects, what kind of genetic changes affects your extract, no? So yun, pwede niyong makita yun. So when you have your cells, you already know the genetic state of your cells, no? So it's visible under the microscope. You'll be able to see what kind of cytogenetic changes occur once you expose your cells with a specific extract. Okay? So you can do that. Also, gene editing, no? Pwede rin yun. Specifically, my current interest kasi ngayon is doing CRISPR studies on CRISPR. Alam, if you're familiar with CRISPR, no, it's a gene editing techniques. So pwede rin yun. This is if you see the image here, no, most cancers, some cancer cells, no, have what we call the complex karyotype. Okay, complex is ganyan na karami yung genetic abnormalities. Ganyan na karami yung genetic abnormalities. Okay, na makikita ninyo. Okay, so gene editing, you can turn and off specific oncogenes or tumor suppressor genes, and you can look at what kind of cytogenetic changes occur. Okay, so one of the possible fields yon. In case of fish, so this is an example of fish for the HER2 neo-oncogene in breast cancer. Okay, so if you want to work with breast cancer, no, so makikita ninyo, no, nung tinarget yung HER2 gene sa breast cancer cells, ang daming copies. Ang tendency kasi ng HER2 neo is overexpressed sila in cancer cells. Okay, so maybe you want to look at, maybe your extract wants to turn that off. Maybe yun yung gusto ninyong aralin, and then you'll be able to visualize doon if you can deactivate or remove or kung ano mang gusto ninyong gawin sa isang specific oncogene, pwede ninyong tingnan yon under the microscope using FISH. Okay, so yun, pwede rin yun. Alright, this is one of the most interesting cases I always show my students now when I teach genetics. This is one example. I want to share this with you, one of the cases I have encountered. Now, this is a case of what we call ambiguous genitalia. Okay? So, ambiguous genitalia is when the baby, a human baby in this case, no, meron siyang genitalia na paglabas niya, you cannot identify kung male siya or female. Okay? Especially in students, no, kung medicine kayo in the future, you'll be encountering cases like this. Pag nag-OB kayo or pedia, you'll be encountering cases like this when you go to medicine, if you go to medicine. So based on this lang, no, based on this image, no, you don't know if this is a male or a female. So that's one of the reasons kung bakit kayo magsa-cytogenetic analysis. You want to look kung male or female yung patient. Okay? Now, what's very interesting in this case is, this is not a case of simple identification of a male or a female. When we did cytogenetic analysis, ang lumabas na karyotype niya is female. Okay? Female siya. We were able to discover na female siya. But the patient has a structural abnormality. So you have your translocation between chromosome 6 and 15. Okay, and then this is interesting kasi this is very rare. This is actually the first documented case of this specific structural abnormality. Okay, before we encountered this, wala pang na-discover na ganitong structural abnormality sa buong mundo. When published, at least, no, wala pang published na ganitong klaseng structural abnormality. So we were the first ones to identify this. Now, interestingly, we want to see kung totoo bang translocation 615 yung patient na to. So we did FISH, fluorescence in situ hybridization. That's when we were able to prove na totoong 6 and 15 siya. Kasi if you look here, no, yung chromosome 6 supposed to be colored green lang, and your chromosome 15 is supposed to be colored pink. Pero kung makikita nyo, no, yung pink napunta dito and yung green napunta dito. So nagkaroon ng exchange of genetic material. Okay? So nakita natin na yung ano talaga, yung abnormality in the 615. Another very unusual fact about this patient, no, most cases kasi ng ambiguous genitalia, they have the abnormality either in the X or the Y chromosome. Most cases, either nasa X or sa Y chromosome. But for this patient, wala, normal yung X niya. Pero nasa ibang chromosome yung abnormality niya. So yeah, that's one of the possible applications ng cytogenetic analysis, no? So yun, interesting. However, sabi nga natin, no, my talk is basically multi-species. I want to talk about the importance of cytogenetic analysis, not only in humans, no, not only in humans, but in other species as well. Okay, so this is just a summary, no, of what we know about the current chromosome numbers and morphologies in non-human species. Okay, so we have your cattle, you have 60 chromosomes in cattle, sheep 54, horse 64, dog 78, ang dami, no? In cats, 38 yung chromosomes nila. And iba-iba yung morphology ng sex chromosomes nila. Meron din silang X and Y, just like humans. Okay? Pero kung titingnan nyo, iba-iba yung centromere positions ng X and Y chromosomes ng iba-ibang species. Diba? So yun. So we have beta-centric, acrocentric, submetacentric, iba-iba rin. Now in plants, no, depende kung your plant is monoecious or dioecious. Your dioecious plants are what usually have the male and female chromosomes. Okay, so mamaya I'll show some examples of that as well. But in plants, mostly ang tinitingnan natin is chromosome number. So titingnan nyo, no, iba-iba rin. For example, rice has 24 chromosomes, corn has 20, cotton has 56. So marami. Sugarcane has 80. Ang dami. Ang dami ng chromosomes. So sugarcane din. So iba-iba, no? And you can think na that's why there are a lot of different applications ng cytogenetic analysis na hindi lang sa human samples. Okay? So whether your interest is plants or animals, marami kayong pwede paggamitan ng cytogenetic analysis. Okay? So, first example ng non-human species na pwede nyo paggamitan ng cytogenetic analysis is for primates. Remember, primates is our closest relative, no, in humans. So, the species Macaca fascicularis, so it is commonly known as the long-tailed macaque. So, we know that these macaques have 42 chromosomes. So, iba, no? 46 tayo, 42 sila. Macaca fascicularis is one of the most common animal models for studying diseases of the human immune system. Okay? When you want to do studies on the human immune system, syempre you have to start from the laboratory, you have to start from cells, then eventually you'll go into animal studies. Now, Macaca fascicularis or the long-tailed macaque is closely related to us, no? Kahit 42 lang yung chromosomes nila, marami silang similarities in humans. That's why they are one of the most commonly studied animal models. Now, even better, no? Macaca fascicularis has a Philippine subspecies. Okay? So, merong subspecies ng Macaca fascicularis na dito lang makikita sa Pilipinas. Okay? So, maybe it has some different genetic differences with other subspecies of Macaca fascicularis. And maybe those differences will result into different responses when you're looking at kung ano man yung gusto nyong i-test. Diba? Remember, a single gene abnormality can result in a different response to whatever you're studying on. So very important to take note yon. Now, isa sa mga common studies, no, which uses Macaca fascicularis is the study on the MHC gene. MHC is the major histocompatibility complex. Okay? So tayo, meron din tayong major histocompatibility complex. Diba? So studies on the MHC gene, which is also present in Macaca fascicularis, can provide data on infectious diseases, organ transplantation, and the development of vaccines. So yan, no? So maraming pwedeng aralin muna sa animals before we test it on humans. And cytogenetic analysis is one of the steps where we can achieve that. Okay. So yun, that's for Macaca fascicularis. So pwede, you can do Gbanding or you can do fish then. And then, you can do that. Okay. Other primates naman pwede rin. You can also do studies on other primates. So in this case, these are New World monkeys. So Pitesha. This is a species of a New World monkey. So iba naman, sila naman, meron namang 48 chromosomes. Okay? So medyo, in terms of evolutionary relationship, no, medyo farther ng konti sa atin ang New World monkeys rather than the macaques. Okay? So if you look at the phylogenetic tree ng primates, no, medyo mas malayo ng konti si New World monkeys. But we do want to see, no, studies want to see kung ano yung genes na meron tayo at meron din sila. Okay? So what the study did, so they used human fish probes. So yung sequences, no, ng fish sa humans, fish probes, yung fluorescent molecular probes, no, what they did was ginamit din nila yon sa monkeys para makita kung ano yung genes na present sa atin at sa kanila. Okay? So pag G-band lang kasi yung ginawa ninyo, no? Pag G-band lang, okay, iba yung itsura nila, no? Pero hindi natin alam kung yung similar itsura ba na yan is yung genes ba na nasa atin is meron din sila. Meron din sila. So pag prinoses mo yung fish, kung ano lang yung magkulay na target ninyo, yun lang yung visible under the microscope. So you'll be able to see, no, na yun lang yung visible. So that's what will give insight ngayon on evolutionary data, kung ano yung genes na na-retain lang from monkeys, from these New World monkeys, na meron din tayo. Okay? Siyempre, applicable din yan sa ibang species ng primates. So if you want to do evolutionary studies, pwede. Applicable ang cytogenetic analysis. Okay? So this time, we can talk about cattle naman. So if your field of interest is agriculture, livestock, you can do chromosomal analysis on your cattle. If you're doing cattle breeding, no? So cattle, we know that they have 60 chromosomes. And then chromosomal abnormalities in cattle naman are often associated with reproduction-related problems. Diba? Whether you're breeding cattle for milk production or for beef production, depende. Kahit anong gusto ninyo, no? May gusto kayo i-breed. Diba, remember, for example, when you want to breed Wagyu cattle, very important yun. Usually, pure breed yung Wagyu cattle, no? And then sometimes, no, because of genetic abnormalities, nagkakaroon ng infertility and nagkakaroon ng early mortality ng embryos. Okay? So, hindi nabubuhay yung offspring. So, maybe sometimes you can look at that, no? Does your cattle have a genetic abnormality kaya hindi nabubuhay yung offspring? So, important yan when doing cattle breeding. So, you can also look at genetic abnormalities using cytogenetic analysis. Okay? Isa pa pala, another example of doing cytogenetic analysis is when you want to study genotoxicity. Okay? Genotoxicity is when your DNA gets damaged, gets broken by specific agents. In cattle, no, an example this is kung yung grass na kinakain nila was exposed to pesticides. Okay? Maybe nagkaroon ng exposure. So what happens is na-expose si cattle sa pesticides. And as you can see here, no, pag tiningnan mo yung DNA ng cattle na yun, sometimes they get broken. They get fragile and they get broken. Okay? Nasisira yung DNA. So these kinds of breakage in DNAs, no, may also result in abnormal offspring and milk production. So pwede rin yun. So genotoxicity studies. Cats, pwede din. May studies din on cats on chromosomal abnormalities, no? So cats have 38 chromosomes. Usually, ang nakikita sa cats is reports of sex chromosome abnormalities naman. So sa X and Y abnormalities ng cats. Okay? So maybe you're breeding cats. Some reason, maybe. So you can see na monosomy X, no? So in cats na males cats, no, nawala yung X chromosome nila, so they have a virilized phenotype, underdeveloped penis, and the scrotum-like structure. So abnormal yung reproductive organ ng cats. Also, you have your translocation X and Y, pwede rin. So you have what we call the DSD or disorder of sex development, underdeveloped. Parang kanina rin, no, similar dun sa ambiguous genitalia ng sa humans. Pwede ring makita rin yan sa cats. Okay? So yan, you can actually test them for cytogenetic analysis. Okay? So that's for cats. Dogs, pwede rin. If your animal of interest is dogs. Kaya lang, ang dogs have 78 chromosomes. Like we said kanina, no, marami chromosome nila. Most studies din ng dogs, focus on DSD, disorders of sex development. And cancers as well. Alam natin na nagkaka-cancer din ang dogs, di ba? So pwede rin nating aralin yon through cytogenetic analysis. Maybe we can, it's actually possible, no, na maybe ang cancer, una nating ma-discover yung cure sa animals. Maybe. Diba? We're still trying to look for the cure for cancer, no? And maybe we can see that in animals before we can see that in humans. There's a possibility. one notable characteristic ng chromosomes ng dogs, no, is they have a high chromosome number, yun nga, no, and a one-arm morphology ng all autosomes. Ibig sabihin, yung autosomes nila, no, yung chromosomes 1 to 38 nila, I think 38 or 38, one chromosome 1 to 38 nila are all acrocentric. Okay? Wala silang metacentric. Well, yung X nila submetacentric. Yun. Acrocentric yung autosomes nila. So, very interesting din yung ano na yun, yung state na yun ng chromosomes nila. Okay? So, that's for dogs. Now, you can also do chromosomal analysis if insects. Sabi nga natin kanina, no? Pwedeng aralin yung samples from mosquitoes. So the Anopheles mosquito, carrier of malaria. So sample of salivary gland cells. So, sabi nga na ba, pwede i-process, no? And then you have 6 chromosomes in mosquitoes, but in Anopheles mosquito, it often appears as 5 arms. Okay? Yung X chromosome nila kasi is polytenized. It only appears as 1 arm usually, no? There are visible high level of inversions in some species. So nagkakaroon ng exchange ng inversion, baligtarang genetic material, no? So you can do studies for vector control. Maybe you want to control kung ano mang yung mutation nila, or you want to see yung if you want to hinder their development, pwede yun if you want to do studies on that. Okay. Aedes aegypti, the mosquito carriers of dengue and Zika virus. So, pwede rin. You can do chromosomal analysis on that as well. So, discovered to have lower gene densities and higher satellite DNA content. So, iba. There are some notable differences between other species ng mosquitoes. This can also be used to facilitate evolution studies. Diba? Malay natin, merong mag-evolve na ibang carrier ng mosquito. May new species na palang mas delikado, no? So we can see that. Pwede nating tingnan yon kung anong pwede na evolutionary patterns ng mosquitoes. Pwede rin yun. In terms of plants naman, in terms of plants, so an example would be hybrid wheat. Okay? So maraming studies on plants na you crossbreed specific plants, no? Between specific species. Okay, so this specific plant wheat species, no, is the Triticolamus. It's the hybrid of the species Triticum and Lamos. Now, this study wanted to see, no, how many chromosomes ang nai-inherit from one breed or nai-inherit sa kabila. Kasi nga, doon mo makikita kung paano magiging viable yung offspring mo. Sometimes kasi, no, specific gene combinations result in a non-viable offspring. Okay? Hindi nabubuhay yung offspring. So you want to see now kung anong genes ang pwedeng makuha sa isang breed at sa kabila para maging viable yung offspring nyo, yung breed ninyo. No? So ito, no, for this example, kinulayan lang nila ng red, yung galing sa Triticum, and kinulayan nila ng yellow, yung galing sa Lamos. Okay? So now they're able to identify how much came from one breed and came from the other. Okay, so when if your interest is plant breeding naman, you can do this. You can test for chromosome analysis ng hybrid plants. Okay, so yun. Rice as well. So rhiza, you can do chromosome analysis ng rice. So in this specific study naman, so rice has 24 chromosomes. They wanted to compare the chromosomes of domestic rice, that's the rice that we know, no, Oryza sativa, and some species of wild rice, in this case, Oryza officinalis. So the study that they're looking at is the bacterial blight resistance gene. Okay, so there are yung wild rice kasi, no, known siya na meron siyang resistance to bacterial blight, and we want to transfer that in our domestic species ng rice. So nakita, no, na in this specific study, no, nakita nila na yung location ng gene na yon nag-iba. It moved from one location to another. In this case, they're still in chromosome 5 ng rice, no, but the gene, instead of being located in the long arm sa wild rice, it was located in the short arm sa Oryza sativa. So it's a different location na. So that will now provide strategy ngayon, no? If you want to do gene editing, when you want to do hybrid plant breeding, so you have to know kung nasaan yung location ng target gene ninyo. So you'll be able to know your agronomically valuable genes. Okay, so yan, an example. Okay, so that's for rice. Beets, Beta vulgaris, the vegetable beet. So there are they have 18 chromosomes and they have 3 types of beets, no? Yung white, green, and red petioles. So magkakaiba yung petioles nila. So they want to see kung ano yung chromosomal differences nila, no? So they saw, no, this specific study saw that there's an asymmetry. Magkakaiba yung length, yung average length ng bawat chromosomes. And then na-identify din nila na yung red petiole beets was a later evolution ng green and white petiole beets. Okay? So kung ano man yung differences nila, maybe there's different chemical components, that's another thing totally, no? But in terms of genetics, ito yung pwede mong ma-identify dun sa species na ganito. Okay? So hemp, even cannabis, very controversial na crop. Alam natin yan ngayon. Diba? Ang daming nagpo-push for legalization ng cannabis. Pwede rin, you can do chromosome analysis on that. Alam din naman natin na cannabis itself has some medicinal components, diba? And those medicinal components are a result of protein expression ng specific genes, no? So, cannabis has 20 chromosomes, and this is an example, actually, cannabis is actually an example of what we call a dioecious plant. Okay? So cannabis has male and female plants. Okay? So meron din silang XX and XY. Okay? Very interesting to note that. And then, yung data na nakukuha nila sa studies on cannabis, no, they actually provide on sex chromosome evolution. So how does the sex chromosomes of specific dioecious plants evolve? So pwede rin yun. Makikita ninyo yung patterns of inheritance. So there are a lot of different things you can do with cytogenetic analysis. Okay. So yeah, that's why it's multi-species. No, these are just some examples of published data on different species for cytogenetic analysis. Marami pa. There are a lot of different organisms you can do cytogenetic analysis on. Okay. All right. So for the last part of my talk, allow me to share some capabilities ng lab where I work in today. No. So I am working with iCell. Okay, so iCell, or the Imaging and Cell Culture Laboratory, we have these specific facilities ngayon, no? So we have the confocal laser scanning microscope with live cell imaging system, meaning we can work with living cells. We'll be able to see cells. You can test your extracts. Now, you can see the live effect of your extracts on cells. Okay, so we also have, newly lang, ano, kung nakita nyo with the presentation of Doc Karen, wala pa nga dun sa list na yon yung machine namin. Very newly installed lang kasi, no, our fluorescence microscope with the cytogenetics workstation. So bagong-bago, kaka-unbox lang namin. So we'll be able to do cytogenetic analysis as well. Siyempre, we have our cell culture rooms where we'll process our samples, and we have our molecular biology laboratory to do accompanying experiments as well. So this is our machine that we have right now. So the ASI Axio Imager with ASI Cytogenetics Workstation. So you'll be able to do analysis for Gbanded analysis as well as fluorescence in situ hybridization. We'll be able to do that as well. So we can do bright field karyotyping. So we can do G-bands, R-bands. We can do fluorescence karyotyping. So may Q-band, R-band na fluorescent, no? And of course, fluorescent in situ hybridization. And syempre, spectral karyotyping. Kaya din namin gawin yon. Kaya din natin i-analyze yon. Spectral karyotyping. So the ASI, Cytogenetics Workstation, can capture both metaphase and interphase cells. Sabi nga natin, no, the cells pwedeng wala sa dividing stage, pwedeng nasa non-dividing stage siya. Pwede yun. So the ASI Cytogenetics Workstation is an all-in-one platform to analyze chromosomes and classify cells. It has high accuracy of automated karyotyping. By the way, this is automated, no? So what we do is ilalagay lang natin yung slide natin dun sa machine. Siya na yung hahanap ng chromosomes. Hindi na natin kailangang manually hanapin yung chromosomes natin under the microscope. Ilalagay lang natin yung slides natin sa microscope. Automated na, robotic yung stage ng microscope namin. Hahanapin na niya kung nasaan yung cells na gusto nating hanapin. Okay? So meron din siyang automated karyotyping. Although sa automated karyotyping, hindi siya 100% accurate lagi, no? Kahit sa machine naman. There's always the human input. Okay? There's always the human input of skill when it comes to cytogenetic analysis. But it gives you at least a starting point, no? So whether it's human or animal, in this case, may mga may mga established patterns na ng karyotype analysis. So automatic bands estimation and auto overlap score. Even if nagpatong yung karyotype nyo, yung chromosomes niyo under the microscope nyo, kaya niyang basahin yon. Kaya niyang basahin yon. Okay. All right. So that's the end of my talk. Take note lang, no, by the way, before I end, take note lang na kami po, iCell is, as of right now, hindi pa po kami fully operational, ano. We are still, sabi nga namin, kaka-unbox lang ng machine namin, no. We are still not accepting samples for processing as of now, no. What we're doing right now, we are still in the stage of collaboration. We are talking with other institutions, possible institutions in the future who can work with us and who can make use of the machines. But hopefully, no, by the middle of next year, hopefully we'll be able to offer this service to all students and teachers and institutions that will be interested. So hopefully by next year, magiging fully operational na kami and we'll be able to offer this. I hope you learned a lot, no, from my talk and what kind of interests you can have, and then you can be able to work with us in the future, no, maybe in 1 to 2 years when we're fully functional. You'll be able, your students, your thesis students will be able to work with us, and then yon. Right? So these are the details. Now again, I'm from iCell, the Instrumentation facility. So that's my email address. If you have any inquiries or questions, I can help you with that as well. And then you can check out the Facebook page and YouTube channel also of DLSU CIF for updates. When we're fully launched na, no, when iCell is fully launched, we'll be posting our updates there. We'll be announcing. Hopefully, we can promote with PhilSci Hub as well then, no? Pag fully launched na kami yung iCell, hopefully we'll inform. So stay tuned sa PhilSci Hub for the subscribers of PhilSci Hub. Stay tuned as well. Hopefully we can announce it, and then we'll be able to collaborate with you guys in the future. Okay. So again, maraming salamat po. Thank you very much for the opportunity, and I hope you learned a lot, and I hope you now have a better knowledge and information on cytogenetic analysis. So thank you very much po sa mga nakinig, nanood.
Ayan, maraming salamat po, Sir Michael, para sa napaka-generous po ninyo na pagbabahagi ng inyong expertise when it comes to multi-species cytogenetic analysis. So, being a chemistry major ako, parang ang dami ko natutunan na nabalik ako sa aking mga biology courses back in UP. So yun. By the way, so siguro po, so we'll give Sir Mike just a minute para po uminom ng tubig. So what I'll do is I'll go over just a few reminders po. Actually just one, kasi we have our last offering, our last web event for this year before po tayo magbakasyon muna, ano. So allow me to share my screen with you, and baka po hindi po ako nakapagpakilala para po sa mga bago po dito sa ating web events. Ako po si Jeffrey Buntin, and I'm the founder of Filipino Science Hub. So I'm based here in Houston, Texas. So welcome po sa inyo. Maraming salamat po sa pagsama sa isa na namang Sabado. And okay, I think I shared the wrong screen. Please bear with me. Ayan. Sakto na ba? Sakto na ba, Karen, yung nakikita ko? Nakikita nyo?
Okay na. Sakto na.
Alright. Okay. So, for our last offering po for this year, we will be featuring Dr. Mary Grace Garjan. So, she's a senior scientist for Eurofins Agroscience Services here in the US. And next week po, December 11th at 10 in the morning in Manila, Sabado po, same time, she'll deliver a talk titled What's in Your Water? Introduction to Per and Polyfluoroalkyl Substances. So, para po sa impormasyon kung paano po mag-register para sa event ito, you may log on, you may actually check out our official website, www.philscihub.com, and then yung registration information po is also on our Facebook page. Ayun, so ito po ang aming last offering. So bali po meron lang po tayong a couple of weekends na magbe-break kasi ano po, kailangan natin yan. We need to be able to recharge. So bali, ito pong session natin with Sir Michael Arnante, I believe, our 60-second web event since we started in May 2020. So, mahigit na pong isang taon ang bilang ng web events na ating nagawa. And we're always very thankful to Salle University Central Instrumentation Facility for partnering up with us and for generously and voluntarily sharing their expertise po sa napakaraming modern analytical techniques. So ito po mga ganitong klase ng trainings or sharings, to be honest po, usually magbabayad po ang mga tao ng thousands of pesos. But then Salle University is generous enough to share their expertise with you on a number of different topics. And I think one very important point na sinabi po ni Sir Michael Arnante kanina na yung multi-species cytogenetic analysis at Salle ay hindi pa po talaga siya fully operational. But then I think what's most important about this talk is that soon enough, lalo na po sa mga guro at sa mga estudyante who are interested or who are very passionate about cytogenetics, in a couple of years, alam niyo na po kung saan kayo pupunta. Ayan. Alright. Sir Michael, ready na po ba kayo sa ating Q&A? So, we'll just alternate po sa pagbabato sa inyo.
Yes po. Sige po.
Ayan. Karen, would you like to gusto mo ibato yung unang question kay Sir?
Sige. Again, thank you, Mike, uli for your generously sharing your knowledge on this topic, no? So we have a question from Mr. Ryan Noviwang. Are there specialized instruments used in cytogenetic analysis? What are some of these? So I guess perhaps a recap of what you have covered in terms of like the advanced instrumentations that are used for this, Mike?
Okay. So in terms of instruments, for cytogenetic analysis, very simple. Kahit simple light microscope lang, pwede natin gamitin for cytogenetic analysis. Like I said, if you do your staining, kung alam natin yung banding patterns, pwede na natin gamitin yon. However, it's easier when you have a photo microscope. Okay, if you can take a picture, diba? The old-fashioned way, actually, ng early years pa lang ng cytogenetics, what we used to do was, diba, you have your slides ng chromosomes, you have your slides, pinipicturean yun. So, pag na-develop na, as in old school photo developing, what they do is, pag pinicturean na yun, ginugupit isa-isa bawat chromosome and then dinidikit sa papel. Okay? Dinidikit sa papel. So, dapat alam niyo yung banding pattern, syempre, ng bawat chromosome. So, that simple lang. Pwede na yun, syempre ngayon, medyo high-end na, Meron ng software. There's software na for cytogenetic analysis para mapadali na. But even as basic as a normal microscope or a photo microscope, pwede na kayong cytogenetic analysis. At least for Gbanding, At least for the normal state. Siyempre, if you're working with fluorescent stains naman, siyempre, you need a fluorescence microscope. Yun na yung medyo higher end. Okay, so yun. So, even if you're teaching, for example, para sa mga nagtuturo ng genetics and biology, very simple po. Kahit pwede niyong gawin sa school yung experiments na yun, actually, If you have a kahit picture lang ng chromosomes, pwede nyo na actually ibigay sa students nyo yun. Sila yung, ganun yung ginagawa namin sa classes eh. Binibigay namin yung picture, sila yung nagugupit-gupit, or sila yung nagpiprint, and sila yung nag-a-arrange ng karyotype. Okay? So, yun na. That's basically cytogenetic analysis already. So, you don't need a high-end instrument to do cytogenetic analysis. Pwede na yung simple microscope.
Ayan, maraming salamat po, sir. So siguro bago po natin ipatay yung next question, we just like to acknowledge po that we had about 75 participants ngayong araw po nito. Sorry po, Friday evening na po dito. So 75 participants po, Zoom and YouTube combined. So maraming salamat po sa inyong pagsama. And then if there's anyone po, if we have participants on Zoom po who would like to ask their questions live, so kindly, we encourage you po to raise your virtual hand and then we'll acknowledge you and para matanong niyo po yung maiboto niyo inyong tanong kay Sir Mike ng live. Okay. JP, would you like to ask the next question?
All right. So from Miss Maria Cristina Ablao, what factors contribute to genetic alterations? Kasi kanina na-discuss ni Sir yung mga types ng mutations sa sa mga genes. Ayan. So ano daw po yung mga factors contributing to alteration?
In terms of genetic alterations, marami po, there's marami po ang nagko-cause ng genetic alterations. For example, in let's say in humans, no, halimbawa, yung mga abnormalities yung mga syndromes, halimbawa po, yung mga Down syndrome, Edward syndrome, yung mga abnormalities po na yun, these only occur by chance. Okay? So nagkaroon lang po ng errors sa recombination. Okay? So may mga ganong klaseng genetic alterations na nangyayari lang by chance. There are also some genetic alterations which are hereditary. Okay, so meron pong namamana, marami po 'yon. Actually, whether it's human, whether it's human or animal, as you can see, sabi nga natin, 'di ba, when you have studies for breeding, whether it's animal breeding or plant breeding, pwedeng makita yung genetic alterations na 'yon. And the way the parents transfer the genetic material to the offspring, there are also contributing factors to genetic alteration. Another major factor to genetic alteration is environmental factors. Sabi ko nga kanina, di ba, I was talking about genotoxicity. We talked about exposure of organisms to toxic substances. Kanina, di ba, I mentioned about cattle being exposed to pesticides. I forgot to mention, or hindi ko lang din na-mention kanina, I was able to perform or assist some students before who did genotoxic studies on humans. Specifically, meron kaming mga populations, halimbawa, mga jeepney drivers na exposed to air pollution. Ano pa bang some examples? Yung mga nagfo-photocopy, yung mga
sila ate.
Machines. So, ate, kuya, di ba? Nag-handle ng Xerox. Alam natin na yung photocopy machines, mainit yon at may ibang klaseng radiation yon. So, tiningnan din natin kung nagkaroon ba ng effect sa chromosomes nila. Nagkaroon ba sila ng breakage? Nagkaroon ba ng fragility yung chromosomes nila? Another contributor to genetic alteration yon. We were able also to do genetic analysis sa mga residents ng isang dump site sa Payatas before. We were able to see kung nagkaroon ba ng effect yung long-term pagtira nila dun sa ganung klaseng environment dun sa status ng repair mechanisms nila. So yun. Surprisingly actually, yung mga nakatira sa dump site na yun, their DNA or their chromosomes were very resilient. Surprisingly, very resilient. So maybe naging kung hindi naman immune, or na desensitize na yung kanilang genetic material, maybe. So yun, very interesting. Kaya marami po. There are a lot of different factors which can contribute to genetic alterations.
Ayan.
Alright,
Thank you.
before tayo...
So ayan talaga, may ano no, marami na Caesar na iba to ng mga research ideas na dun sa mga sinabi niya na examples.
Yan. Oh, maraming mapulot. Ang maganda kasi doon kay Sir, pinasadahan niya napakaraming species, no? So,
Exactly.
kung hindi niyo po kayo nakapag-take ng notes doon, balikan niyo lang po sa YouTube kasi recorded naman to. Pero before natin nabato yung next questions, kasi we have quite a few. Ano lang po, advertisement lang din po ulit. So today po, so at 1 p.m. in Manila, we have our podcast featuring our individual contributors and interns. So yung mga college students po who are contributing to Filipino Science Hub. So for today's episode po, we will be featuring Gerald Gayona and Michelangelo Lupac, and yung topic po nila is about yung dilemma ng karamihan po ng nagpipick ng major for college, and yung kapag nandun ka na sa college program mo, kapag medyo na-realize mo, okay, yung passion ko nasa ibang area pala. So should I shift or should I not shift? Or even after ng pagkatapos yun ng degree, say ako, nakatapos ako ng chemistry, ipapractice ko ba siya or should I pursue another area of passion? So we have a very good resource person for this. So we have Dr. Connie Giba. So she's actually currently one of the leading Filipina experts when it comes to mycology and taxonomy. So she's based in San Antonio, Texas. Just to give you an idea po ng kanyang background, she graduated with a medical technology degree from Salle University. When she went to UP Los Baños, she pursued masters in veterinary medicine. And then after that po, she also pursued a couple of other areas of expertise. So dito po nyo makikita how Filipinos and like how an individual can actually navigate over a number of different areas, but still at the end of the day, like come out being very highly successful and world recognized. So ayun po, it's at 1 p.m. po. We're live streaming this po on Facebook. So kapag after po ng pananghalian nyo, if you have time and you would like to check out her story, this is a very good piece po to watch out for. Okay, now we're going back to our question. So Karen and JP, whoever between the two of you is next. Please do so.
Okay.
So, all right.
Sige. From Ryan Huang again. So, ang tanong niya is, are there other human cells aside from RBC which cannot be used in cytogenetic analysis? Is polyploidy always lethal? So, yun, Mike.
Okay, so these are two different questions, no? Sige, isa-isa natin. Human cells which cannot be used for cytogenetic analysis. Maraming skin na pwedeng gamitin. Actually, maraming cells na hindi pwede. It's not more so of a hindi pwedeng gamitin, no? It's more so na it's much more difficult to process. Okay? Technically, any cell, no, that has a nucleus, that have chromosomes, are pwedeng gamitin sa cytogenetic analysis. However, remember that ideally, we have to get them at the dividing stage ng cell cycle. An example, one of the things we do for cytogenetic analysis before was for babies. Halimbawa, I used to work in a hospital, no, for babies na miscarriage. Okay? So nagkaroon ng miscarriage, no? Minsan tinatest namin yung fetus, yung baby, after alisin sa nanay, no? Tinatest natin siya for cytogenetic abnormalities. Tinitingnan natin if yung reason ba kaya nagkaroon ng miscarriage is a genetic abnormality. Now, syempre, walang blood cell dun. Medyo wala pa halos blood cell yun. Pag fetus, wala pa halos blood yun. Sometimes we do test from the skin, the other parts ng tissue. The only problem is sometimes mahirap nang hanapin kasi yung dividing stage ng cell cycle. Okay, mahirap i-timing yung cells na yun. So that's why we do recommend that the ECS is really white blood cells. Okay. But technically, any other cell na merong nucleus at may chromosomes, pwede. One common actually is yung swab cells from the cheeks. Pwede rin gamitin yon. Okay. So, but yeah, it's much more difficult lang to process other cells. Kaya discouraged na gamitin yung mga yon for cytogenetic analysis. And for your second question, is polyploidy always lethal? In developing organisms, most of the time, yes. Okay, in developing. So kung ipapanganak pa lang, whether it's human or an animal, mostly ipapanganak pa lang, i-develop pa lang, most of the time it's lethal. Bakit? Kasi nga, sa development pa lang in the development pa lang ng organism, no, abnormal na yung number. Ibig sabihin, napakarami ng genes na abnormally expressed. Okay, napakarami ng genes na abnormally expressed, and yung pag-develop pa lang ng organism hindi na viable. So mamamatay na yung offspring. Now, in terms of, like I said kanina, no, cases of polyploidy that we can also observe in cases of cancer. Okay? Technically, you can say it's lethal kasi it's a case of cancer, no? But it's not immediate lethal. Siyempre, di ba? I mean, there's treatments in specific types of cancer, maybe at least to delay the progress. So, yun. But the general rule is, the more polyploid a cell is, the more genes are abnormally expressed, the higher tendency talaga na magiging lethal. Okay, but not all the time, syempre. Or not immediate rather, not immediate. So, yun.
Ayan. So, naalala ko dun sa sinabi ni Sir yung cheek cell, no? I don't know, Karen and JP. Sa UP pinaganon tayo, no? Either Bio 1 or Bio 2.
Oo, parang meron.
Sa microscopy, no? Pero I don't think we actually did cytogenetics, no? Medyo ano lang yan. Nakarelate lang po kami dun sa sinaswab yung loob ng pisngi.
Actually, sorry.
No. Go, Karen.
Yun, actually, I'm just really amazed at like what Michael is talking about right now, no? Kasi makita mo how this yung ramifications of these changes or alterations from yung cascade nung or yung how these small or slight alterations can actually have very large ramifications or magnified effect dun sa organism. So I'm just really amazed with that one.
Oh, at the molecular level, how it translates to, like the macro structure of an organism.
Yes.
Napaka-impressive. Biology is really it's really amazing, but then also quite scary.
Mga chemist takot sa biology, no?
Yeah. I mean, kasi chemists po like us, like GP and Karen, we're dealing with simpler, much more controlled systems. But then when it comes to biology, kasi it's very difficult to make correlations or even to visualize things. So kaya po sobrang bilib din po kami sa mga biologists kasi mahirap po talaga yung ginagawa nila. And I don't want to generalize, no, pero I think when it comes to yung complexity ng research, I bet it's much more difficult for them.
Pero hindi tayo takot sa mutagen.
Yeah. Ayan, kami po yung mga exposed sa mga kung ano-ano. So, siguro magpa-cytogenetic analysis na rin tayo kay
Magpa-test ako kay Mike.
Yes. And kami ni JP, I bet that we have handled a bunch of different materials na majority ng mga tao ay
carcinogen.
Yeah. Opo. So, alam niyo na po. Anyway, so going back to our questions, so we still have a few to cover. Ayun.
Okay, I guess this is from YouTube. From Manolita White, why is karyotyping performed in the metaphase stage of mitosis?
All right. I explained this a bit kanina, no, but I'll explain it again. Remember, in the phases of the mitosis, no, in cell cycle, the prophase, metaphase, anaphase, telophase. Now, in prophase, at the start of mitosis, no, that's where the condensation of the chromatin material starts. Okay, when it's still in the interface, no, hindi pa compact yung structure ng DNA or yung chromatin or chromosomes, hindi pa sila visible under the microscope. They're visible, no, but they're in a very spread out form, yung chromatin material. Now, upon prophase, that's when it starts condensation. And when the when metaphase occurs, that's where the most condensed form of the chromosomes are. Okay. Now, when the stage of mitosis goes to anaphase, it's hard to study na cells. It's hard to study chromosomes at the anaphase stage now of the cell cycle because the chromosomes have already split. Okay. Now, because they have split, you might mistake them for polyploidy because you thought it's 2 chromosomes, no? Kasi nag-split na nga siya. But in reality, it's only 1 because it's already in anaphase. So that's why we need to study cells at the metaphase stage. That's the stage where the most condensed and the most visible form of the chromosomes are.
All right. Okay. Next question.
How does karyotyping detect, from Manolito White again, how does karyotyping detect aberrations in chromosomes? What is the karyotype of Down syndrome and Turner syndrome?
So yun yan,
no?
Mga
trivia questions na to.
Okay lang mo, it's okay. So when doing karyotyping nga, no, di ba? You detect aberrations by first knowing what is the normal state. All right? What is the normal state of the karyotype of the organism? So in our case, humans, we have 46 chromosomes, XY in males, XX in females. Now, any change from that is now an aberration. All right? For example, in Down syndrome, you have cases of trisomy 21, meaning normally we only have 2 copies of chromosome 21, but in cases of Down syndrome, they have 3 copies. That means all genes in chromosome number 21 are overexpressed. That's what causes your abnormalities. In cases of Turner syndrome naman, so these are females, no? But instead of having 2 copies of the X chromosome, they only have 1 copy. So that's why they are underdeveloped females because you need 2 copies of the genes in X chromosome for it to be a normal female.
Okay. And then I think yun namang question about yung karyotype ng Down syndrome na discuss naman na yun, sir, kanina. All right. Let's move on to the next question then. Okay.
Okay. From Ms. Hannah Alcanzar from Davao, I would like to ask if there is an SOP for anti-cancer studies of natural products. Should my sample be an isolate or is it okay if it's a crude extract only?
Ayan na. The question.
Actually, baka si Doc Karen ang mas rate sumagot nito. No?
You know, so actually, I'm not sure whether it's a question for my...
Sige, I'll answer after you, Doc Karen.
Yes. Okay. So, kapag natural products, of course, you're expecting that you would have hundreds to even thousands of components, no? So, if you want to look at anti-cancer effects of particular components, in a way, it's also good to look at a more complex view of the natural products kasi minsan meron ka mga synergistic antagonistic effects dyan eh. So if in some cases yung kapag in-isolate mo na yung active component or yung specific compound, minsan nawawala rin yung activity nun because yun nga there is the synergism or synergistic effect in place for it to really function well. So in some cases, it's good to have a clean, isolated, or purified extract. In some cases, you can also look at hindi siguro crude, but perhaps different fractions of your extract for the studies. Yun.
Okay, so siguro yung point ni Dr. Karen dito is sometimes po, yung effects that you would actually see is not just a function of one particular component, but essentially a sum of the function of a number of different components po. So sometimes extracts could be good, sometimes medyo pure component or like narrower composition of components. So siguro po ito ang message dito is it's very difficult to prescribe which is which kasi it varies from case to case. So yun. Sir Mike, do you have anything to add?
And then to add lang, no, it's better to know if what's important, I think, no, is better to know what kinds of effects your extract would have on specific cancer cells. Are you looking for your compound of interest to basically kill your cancer cells? Are you looking to change them? Are you looking to change their activity? Are you looking to hinder their growth? So yun siguro, no, in terms of the cancer cell component, no. I mean, hindi po ako chemist, so hindi ako expert sa extracts. That's why I'm saying na siguro in terms of the cellular and cytogenetic component, no, it's better to identify muna what do you want to see in cancer cells. Ano yung hypothesis ninyo na mangyayari sa cancer cells because of your extract. So very important to see yun.
All right. I think we're down to our last question.
Okay, from Miss Maria Cristina Ablao. What causes errors in cell division despite the fact that there are already checkpoints in the cell cycle that detect errors or damages and provide time for the cell to repair before DNA replication?
All right. So in terms of cell cycle checkpoints, no, alam po natin na all cells, we know that all cells have the capability to really repair your errors in DNA replication. We already know that. However, sometimes, no, in cases, halimbawa, no, in our experience, in cases of genotoxicity, if there is too much broken DNA fragments, the cell can only catch up so much. All right, there is a limit po. There is a limit on how much can a cell can repair your DNA fragments. All right. I mean, I'm not exactly sure of how much. I mean, there's no specific number po, ano, but the point is the cell can only repair so much. Okay, so kung masyadong marami po yung errors in DNA replication and hindi na nakakaya ng cells yun. Some even, no, there are some mutations na hindi nare-repair ng cell cycle checkpoints. For example, yung trisomies, nababypass nila yon. So there are mechanisms regarding that, and that's why yun yung nakikita natin sa cytogenetic analysis na hindi lahat ng errors in DNA are being repaired. So yun.
Ayan. So siguro we can entertain a couple more questions kasi meron po mga bagong pumasok na I think are worth answering. We can finish in just like a couple of minutes. So this question is from Cliff Moises Florentino, so from Zoom. So sabi niya po, sir, so upon knowing the genetic disorders upon karyotyping, are there already ways by which we could still correct these numerical or structural abnormalities? So meron po bang intervention?
Okay, so in terms of intervention, no, dito na ngayon papasok yung concept ng genetic editing. Okay, now in the field of genetic editing is still a very, very young concept. Alam natin, CRISPR was recently discovered lang, no, relatively recently discovered lang. So in terms of halimbawa nakita na natin yung genetic abnormalities sa cytogenetics, no, most likely we will not have a way to correct that. What's important to know is you already know your cells have this specific genetic abnormality. And that genetic abnormality corresponds to a phenotype. Ibig sabihin yung characteristic ng cell or ng organism na yon. What we can do is we can isolate, no? For example, in cancer cells. One of the problems in doing cancer studies is specificity. Alam natin ang biggest problem with cancer treatments is you target not only the cancer cells but also the normal cells. So, 'di ba, may mga treatments tayo na kaya very very intense yung side effects sa katawan ng isang cancer patient ng treatments. So the newer way of studying cancer treatments is we want to see if we can develop a treatment na ang targetin lang natin is yung cells lang na meron yung specific genetic abnormality na yon. So rather than treating, no, halimbawa, siyempre, in cases of Down syndrome din, hindi natin kayang ibalik yon, hindi natin kayang i-revert, no. Wala pa tayo sa level of technology na yon. But what's more important is being able to identify what genetic abnormalities an organism has and trying to see kung ano yung pwedeng modifications natin gawin, most likely on a phenotype level muna. Okay? Hopefully, eventually in the future, we'll be able to develop technology pag kaya na nating mag-gene editing, no? So we'll be able to see kung kaya natin i-revert back yung ganong klaseng abnormalities.
Alright. So, okay. So, maraming salamat, sir. So, last question. This is from Mary May Michelle Lumaban. Her question says, how can we contact you, sir, if we need consultations in cytogenetic research? And yung second question, a follow-up question is, are there facilities available for other schools doing research?
Okay. So my email address po was in the presentation. So you can contact me po there. Currently po, no, I am, as I said, I am working from the Emerging and Cell Culture Laboratory, although we are still not fully functional, no? But eventually, just like the other facilities po ng CIF na fully functional na ngayon, eventually when we do launch and we do fully operate, it will be available po for other schools. Depende if you want to do a collaborative research, if you want to do your students to do thesis, or kung gusto niyo lang po mag-send ng sample, eventually we will be able to work with all those kinds of collaborations. Sa ngayon lang po, hindi pa po talaga, as in, like I said, kabubukas lang po namin ng machine. So we're still in the process of developing the protocols, even the prices, yung mga presyo po ng paggamit ng machine. We're still in the process of developing that and yung mga kung anong klaseng mga collaborations ang pwede namin gawin. Hopefully, by middle of next year, we'll be able to fully launch and we'll be able to work with other schools. Hindi lang po kami pang DLSU. Hindi po. We welcome all other institutions that are interested in doing these kinds of research.
All right. So next year, pababalikin natin si Sir Mike.
Sige po, pwede po. If anyone's interested, pwede po.
All right. And then we would like to ask JP to do the honors of presenting this. Yeah.
Okay. So in behalf po of all the participants present here on Zoom and on YouTube, and all of us at PhilSci Hub team, we would like to present the Certificate of Appreciation to Mr. Michael Arnante of Salle University Central Instrumentation Facility for delivering the webinar Multi-Species Cytogenetic Analysis, organized by the Filipino Science Hub and held via Zoom and YouTube Live on the 4th day of December 2021. Signed Dr. Jeffrey Bungkin, PhilSci Hub founder and CEO, and yours truly, Vice President, JP Onya, Vice President, Filipino Science Hub. So, sir, sobrang thank you po. Maraming salamat for being with us today. It's such a pleasure to talk to Sir Michael Arnante kasi talagang we're listening to an actual expert on cytogenetic analysis at talaga namang sobrang information-packed yung kanyang presentation this morning. And speaking of certificates, I've already shared here the link for the certificates of participation. Just 1 minute until I share it to YouTube. So ayan po, kindly check the link for your certificate of participation and allow us for like 7 days to issue all your certificates kasi by batches po ang pag-issue namin ng certificates of participation. So, and after 7 days po, kung hindi pa dumadating, then that's the time that you could contact us for the follow-up.
Ayan. Sir Mike, maraming salamat po. And then, Karen, as always, ikaw ang bridge between DLSU CIF and then Filipino Science Hub. So, Ito po, yung last and final installment po nitong webinar series na ito will be featuring our very own Dr. Ana Karen Lacerna. So January 15th po yan. So bakasyon lang po tayo ng ilang linggo muna. And then watch out for Karen. So she'll talk about liquid chromatography, instrumentation, and its application in natural products research. Tama, Karen? Yun ba yun?
Yun, yes. So, yun, kita-kita ulit tayo next year on the last installment or last installation of the webinar series ng DLSU CIF at PhilSci Hub. And hopefully, more to come.
Meron pa po.
Marami pa yan.
Meron pa po kami mga workshop on sample preparation and essentially more practical guides on how you can actually prepare samples or carry out certain research or experimental steps that would help you more efficiently treat your samples para pag nag-send po kayo sa laboratory facilities like De La Salle CIF, handang-handa na po. Kumbaga mas madali, mas mabilis nang ma-process and then kumbaga magiging confident po kayo sa magiging result. So this is also again another aspect. These are also other contents that De La Salle University will be generous enough to share with all of you. So hindi lang po sa pagtanggap ng sample, tuturuan din po kayo kung paano mag-prepare ng sample. So watch out for
it's really very important, no? Because for us, mag-analyze lang kami, but you know, the integrity of your results will be coming from the integrity of your sample. So very important talaga na ma-prepare niyo yung samples ng maayos. So yun, hopefully in the next year, we'll get to share more on that.
Wag po kayo magpapadala ng halaman at ng dahon at sasabihin ko po ay paki-ano nga po. paki-detect, paki-isolate nga po at paki-analyze kung ano po ba ang active material dito. So, ang goal po ng De La Salle University CIF and then ng Filipino Science Hub at the same time is to enable you, to give you the capabilities and the skills para po prepare kayo ng mga materials the right way. Lalo-lalo po sa mga guro who are training students, it's very important that they capture those skills. So ayun, maraming salamat po uli sa pagsama. Karen would like to invite Sir Mike for a very quick debriefing session lang. So I already sent the link if you could forward that.
Okay, sige.
Natin pong mga audience members, please enjoy your lunch po. Maraming salamat. And then if you get the chance po at 1 p.m., join us for SciTalk with Dr. Connie Gibas. Ayun. Maraming salamat po and see you next week for our final offering. This year, grabe. Ano po, naka-isang taon na naman tayo. So maraming salamat po sa inyo and we'll see you again next week. Ingat po.
Thank you guys. Maraming salamat po.
Have a good weekend.
Thank you everyone. Thank you.
Have a nice weekend. Thank you, Sir Mike.
Thank you, Mike.
Karen.
See you in the